next up we have Roy Goodacre, who's Professor of Biological Chemistry at the University of Liverpool. Roy's research interests are broadly within analytical biotechnology and systems and synthetic biology. He helped establish mass spec-based metabolomics for long-term studies and employed metabolomics for clinical and plant studies, as well as understanding microbial systems. He helped establish the Metabolomics Society, is editor-in-chief of the journal Metabolomics, and sits on the editorial advisory board of 4 other journals. So Roy's talk is Metabolomics by the Numbers, Lessons from Large-Scale Phenotyping. Over to you, Roy. Thank you.
Hello, I'm Roy Goodacre from the Department of Biochemistry and Systems Biology at the University of Liverpool. And I'd like to thank the organisers for giving me the opportunity to tell you about our work, in particular, some of the lessons that we've learned from large-scale phenotyping. Now, I do feel like a little bit of a fraud talking at a breath conference when I'm not going to be talking about breath at all, but about our work in blood. But I hope you'll see the sorts of things that we've learned along the way, and hopefully these would be able to be applied to breath analysis. So I'm going to tell you 2 stories. Okay, the first is how we set up HuSaMet, which stands for Human Serum Metabolome, and how we went from sample presentation to analysis. And then the second is where we took that kind of protocol and applied it to look at aging in the elderly. Okay, so let's have a look at the first thing. So I should start off letting you know about the co-authors, okay? And what I've done is I've highlighted underneath various people the sorts of skill sets, whether they're basic analytical scientists, basic computing scientists, clinical or industrial. And you can see here, for example, Rick Dunn, who led this work, was an analytical chemist along with Paul Begley, who did a lot of the work. Okay, Wang Chan and Dave did a lot of the chemometrics, and then you can see quite a lot of clinicians were involved in this work. And there's also industry from AstraZeneca and GlaxoSmithKline. And this was funded predominantly via BBSRC and MRC. And the idea was try to look at molecular phenotyping in the UK population. Okay. And what we use here is we use derivatization-based methods for GCMS, and then we use reverse phase LCMS directly on a serum extract. We use both these methods because they complement one another. One looks at central carbon metabolism, and then the LC looks at more lipophilic material. When we did this work, we have 1,600 metabolites in a GC library, so that we know that we get level 1 of MSI identification. Okay. You could ask the question, is the technology stable? And when we started this, we of course thought it was, but of course it's not. Okay. And the reason for this is that mass spec and chromatography drift. When you're doing this work, when you're using derivatization chemistry, you're basically having molecules interact with the solid phase on the chromatography. And this will be just the same for breath analysis. And of course, the mass spectrometry drifts. So we weren't particularly surprised at this, but you know, it does need to be overcome when you want to look at in the order of hundreds or thousands of samples. Okay, and what we came up with were the idea of using quality control samples, and this was published by Paul in 2009. And basically, a QC is a biological QC, and you take an aliquot from all the samples in the study. You make a pooled QC, and then you make small aliquots that go in a freezer, and you run them regularly through the analysis. Okay, and it was important that this pooled QC was biologically relevant. And the reason for saying that is that if we do an analysis with pooled QCs, which are these magenta samples here from all of these samples, and compare it with Sigma Aldrich's serum, you can see that this QA, as we would call it, quality assurance, looks nothing like biology. Okay, so this is not going to be very good for looking at system parameters and for correcting drift in an instrument. But what you can see here, for example, are these QCs lie more or less in the center of these samples where they were generated from these green, these real samples. Okay, and so we've wondered what we could then do with these QCs. And the idea is that you can use those to look how well your instrument has done. Okay, the QCs should always look the same, but you could also use it for signal correction. So here, for example, is the analysis order. So this is 12 months or 18 months, depending on whether it's GC or LCMS for this particular experiment. And this is the signal intensity for a single metabolite that you've extracted. And basically you can look at that metabolite over all the samples that have been analyzed, and you can see these are little batches here. Okay, but you can certainly see that when we have an annual instrument maintenance, okay, that we get a break in that kind of trajectory and a very big change. And you would not really be able to analyze samples before the maintenance and after. And you can see, as I said, the drift in this particular analysis. Okay. Now, the use of the QCs, which are these red ones, are that they're run quite regularly. So we start off by injecting 10 QCs, a blank, then column test, and then our samples, and every fifth or sixth sample is a QC. And so this is now nominally identical material. So the response in the instrument should be the same. So basically what you can do is you can take those QCs, do maths on those. In this case, we did LOIS, which is a sort of locally windowed optimization or warping the signal. And you can now bring the QCs down to a set level and the samples around them because you do this locally. Okay. And so that's one way in which we develop that. We did this, published it in Nature Protocols. It's been very well received. This is 9 years old and it's already been cited 1,300 times. Okay. And if you're interested in that, and maybe if you can get QCs for breath work, then you might like to look at this paper that Dave Broadhurst, who really wrote that algorithm, came out on guidelines. It was published a couple of years ago in Metabolomics. I can't stress that these things are important. I know we might go on about citations, but this has been cited 150 times already in a couple of years. And it basically just pictorially shows again the same process. So here's the QC for that particular metabolite, okay? And after correction, it brings it into a normal range with samples around it. What you then have to do is do batch correction, okay? So these are your batches of samples where you've done this little correction, and then you need to stitch these together, okay? So that is all explained in that particular paper, okay. Obviously, the question is, for our analysis, did the QC work? Okay, we measured a lot of samples. Okay, and what we did is we took the data after correction. We did discriminant analysis, and here we tried to separate the 10 different batches that we'd analyzed. And basically, it did work because we could not see any batch effect, and we certainly could not see time within that particular analysis. Okay. And this correction worked for the XCMS data, also worked for the LCMS data. We did LCMS twice, one in positive ion mode and one in negative ion mode. Okay, so we basically have a pipeline where we have SOPs for how you collect blood, okay, how you quench and extract, and then how you do the mass spectrometry in terms of XCMS and LCMS. And hidden underneath a lot of this is all the data processing that occurs, and I'll touch briefly on that. What I'd like to do is tell you about the Husamet project. So we set up this pipeline. We then analyzed 1,200 subjects. This took 18 months. Okay, so certainly you're going to see a lot of drift in instruments, but we've just seen we've been able to correct that. Okay, and we measured samples from Stockport Primary Care Trust, GlaxoSmithKline, and the European Male Aging Study. Okay. Now, the first thing we did is looked at the LC data, and I'll just play that again, okay? And you can see here the most positive correlation that you're seeing here are 2 ions. This is the sodiated and protonated glucose in electrospray ionization, and it correlates, okay, with serum glucose. This is not astounding, but it's useful to know that we do actually get the correlation with glucose, okay? So clinical measurement versus mass spec, where we have not targeted the analysis for glucose, okay? We've done a general extract and a general quench, and the extract is in methanol, okay? We do the same thing, correlation based again on 1,200 individuals, and look at GC. So these are the clinical parameters. Because we have 1,600 things in our GC library, we can name them. Okay, you might see things that say and or, that's because we have ambiguity for those particular matches. But then we can look at correlations. And if we look at this area here, okay, we find that high density lipocholesterol, okay, is correlated with all the fatty acids, okay, and that's a positive correlation, and that's kind of known, okay, that's sort of lipid, those are lipid issues. If we look at other metabolites, for example, this metabolite here, 4-hydroxyphenolic acid, that's of microbial origin, okay, and when we started seeing this, and remember that these data were being generated 8-9 years ago, okay, that this was a surprise, but of course nowadays it's very regular to know that your serum is awash with microbial metabolites. It also has drugs in it, so we also identify naproxen, salicylic acid and paracetamol, even though we ask these individuals not to take over-the-counter drugs. Okay, and the last one we can identify here and just say a little bit about is caffeine, so this is from food or drink, and quite interestingly in this study, although we asked people not to fast before giving a sample, it's possible that some people had a cup of coffee before they gave a sample, because the variance in that measure for that population was 400% relative standard deviation. Okay, and there are many molecular phenotypes. If you're interested in the paper, it's there, and it'll appear again at the end. And I'll just talk a little bit about gender and a little bit about age, but we also look at things like BMI, smoking, blood pressure, okay, and they're reported in the paper. Okay, so what about the data analysis? Well, there's a lot of data, as there is with breath analysis, okay, and the metabolomics community seems to be obsessed with partial least squares discriminant analysis, and so here's our chemometrics modeler. And we would like to persuade people not just to use PLSDA, but to use things like random forests or support vector machines. And in doing so, actually, we published a tutorial a few years ago now, 5 years ago, being very well received. And it basically is entitled Metabolomics in PLSDA, a marriage of convenience or a shotgun wedding. And really, all that paper did was tell you that there were different ways of identifying, and discussed some of the nuances amongst those. So when we come to do our feature discovery to find out which metabolites are important, we actually do use PLSDA, but we use random forests, and we also use support vector machines. And then we look at committee voting, which basically means we look to see how commonly seen a particular metabolite is. To do that, we do bootstrapping. We typically resample our data 1,000 times. And what that basically means is that you generate a training set with just about 60% in, and the test set with 40%. Okay, and the results that we look at here are from a null distribution. This is when we do permutation testing. And here is a real distribution. This is actually infrared data of single cells looking at control versus a dual therapy, where we can get identification as to whether a cell has seen a drug or not and how it's responded. Okay, but this is what we do with our metabolomics. Okay, we look to see what sort of power we have. So in this case, we have 95% power in this analysis. In this analysis, we only have 80% power. And if you look at the overlap here, then you can start to say how many times, let's say, a real model overlaps with a null model, and that gives you some idea as to the robustness of your analysis. So for the Husamet data, we did a similar thing. We did a bootstrapping analysis where, let's say, we were looking at age or looking at gender. We then also do univariate analysis. We do QQ plots, check for normality, maybe log 10 transform to make things look normally distributed. And then we use ANOVA, and we do an FDR correction, because we are doing multiple testing. And at the end of that, we look at common features, okay, from the multivariate analysis and the univariate analysis. And because all of these methods have very slightly different ways in which they look at the data, whether it's a linear classifier or a non-linear classifier, an ensemble type classifier, you just look at peaks that are seen by all of those, and then they are likely to be more robust. So let's have a look at that. We look at aging, And we find, for example, citric acid here increases as you age, both for females and males, but it starts off higher and stays higher for females than males. It's very interesting. This has been seen before. Okay, and this is due to dysregulation and basically sloppiness, if you like, in your mitochondria as you age. Okay, so you generate excess citric acid. Okay, you can see the ridiculously low p-values here. This is nothing to get excited about. Remember, we have 1,200 people in this population, so we're able to approximate the mean for an ANOVA test very well in this particular case. We look at gender effects. So, for example, here's methionine sulfoxide. It's significantly higher in females than males, irrespective of age, and that is a marker of oxidative stress. And we can see things that people have seen previously, and then we see things that haven't been reported before. And quite interestingly, caffeine is significantly higher in females. That could be drink, or of course it could be food consumption. It could be eating nice chocolates. And then you can look at combinations of gender and age effects. So here, for example, we see tyrosine increases quite dramatically for females as they age, less dramatically for males. And if we look at tryptophan, this basically stays the same for females, but it actually drops as you age in males. And this is well known. This is due to muscle mass turnover. Okay, and these are the age bracket, age criteria. But as you can see, it's very nice to be able to show these and see that they agree with what's been seen in the literature. Okay. Question is, how fast did we get there? Well, we spent a lot of time optimizing the process, but we also spent a lot of time learning. And so actually we got there a lot slower. And you could ask the question, well, why was that? Basically, it took about 6 years to learn how to do metabolomics properly. And that's because the human is basically a superorganism. Okay, there are lots of things that affect the human. There are things that you can't change, like age, diurnal cycle, reproductive status, and things like that. There are also extrinsic factors that are modified. So nutrients, non-nutrients, drugs, okay, things that you're exposed to, level of physical activity, microbiome, and mental status. So these do need to be considered. And if we think of man as a Vitruvian man, if you want to measure him or her, then it's very complex. I told you that we saw microbially derived metabolites, we saw drug metabolites, we saw nutritional metabolites, as well as human ones. So the system is really quite complex. So to do it properly, if you're going to do case control studies, then you need to control the diet, you need to control the sampling time, look at diurnal rhythms, think about estrous cycle, and you need to have very carefully matched controls. It's often not easy when your subject matter is Homer. You might tell him not to drink alcohol, not to eat naughty foods, okay, and to do more exercise, but he doesn't. And that's the reality when we do metabolomics, we do breath analysis, when we think about going into general practice. We need markers that are robust enough that are not affected by our nutritional status, by our microbiome and things like that. We could ask the question, is size important? Okay, this little map here is just meant to remind us that there are rather a lot of us on the planet. Okay, and in our study, because we measured 1,200 people, what we could do is we could sub-sample these, okay, and we took 100 bootstraps at different sample sizes, and then we asked the question, how well were we able to do those predictions? Okay, so if we only take 50, we're around 75% accuracy, let's say, for BMI. Okay, but you can see these numbers increase as you sample more and more. And so if you take this as a sort of point at which the curve flattens, okay, this is where we might reach the so-called central limit theory in the statistical literature for this population. Basically says you need at least 300 subjects per class, okay? So size is important. And we review this, Drupad and Kat Hollywood wrote this review in New Horizons in Translational Medicine a few years ago. And at that time, there were only 4 studies that really had over 100 subjects in each case control class. So when you're doing breath analysis, you do have to realize that your instrument variability is there, your subject variability is there, and so therefore you do need to power the analysis by measuring more subjects. Otherwise, you basically are likely to find things just by chance that don't actually add up. So that's it for the Hughes-Met. So that's how we set up our serum metabolomics, and basically we do this analysis for everything. You can see the little picture here has Homer, but it also has bacteria and it has plants, and any sample goes through exactly the same procedure where we have standard operating procedures, we have QCs involved. We then applied that to look at frailty in aging populations. Okay. This was funded by MRC. We called this Frailty and Resilience and Equality in Later Life. The lead author here is Nick Rattray, who's an analytical chemist. Okay, and again, just like the previous sort of study, this is just to highlight that we have clinicians involved, clinical statisticians. A lot of these people like Gundo and Chris and Bram and Tarani were involved in looking at socioeconomic factors in this particular study. And then we have, basically a mix of basic scientists and clinical scientists, which is what we needed. So I'll give you a couple of definitions. I'll talk about the English Longitudinal Study of Aging, Frailty, and the Rockwood Index. So ELSA is basically a longitudinal study. It's looked at people over the age of 50 and then measured them several times. It's basically a 2-hour questionnaire. Looks at health, disability, looks at how long you can balance on one foot, how often you can sit down. Okay, and it's been done over the last 15 years. This slide's a little bit out of date. It's been going about 18 years now. So it's a great resource where the same people have been followed as they age. So frailty is really related to this Rockwood index. There's no real kind of definition as to what frailty is. If you watch old people walk down the street, you'd say whether someone looked like they were resilient or they were frail based on how they were moving. But biochemically speaking, it's very difficult. And so Ken Rockwood came up with this idea of taking these 100 questionnaires and physical measurements and putting them through an algorithm that basically comes up with this Rockwood index. And for our population, this is just under 1,200 people we analysed at an early wave. Okay, and you can see that most of them actually are resilient. Okay, and there's a few people that have a kind of frail phenotype. Okay, so the first analysis we did was on wave 4. Okay, and we measured nearly 1,200 people again. We did PCDFA, again, linear discriminant analysis algorithm. We separated into 4 groups, okay, and we said, can you separate them? And we bootstrapped this, and this is the results of the test set. And if we look at this, we can separate out non-frail from people who are pre-frail and people who are more frail, okay. And because we had an imbalance, when we then went and did our sort of knowledge discovery, we adjusted for this, and we made sure that we weren't overlooking, at people in the pre-frail category. And you can see this is done on LCMS in the positive analysis. GCMS for this data set did not work very well, and we had a big problem in the fact that identifying features. This was done on an Orbitrap. Was very hard because we didn't have a library like we have for GCMS. So we use Mummichog within XCMS, and XCMS is a deconvolution software. And basically the way that this works is it identifies a feature based on just accurate mass alone, and it then enriches this particular pathway, let's say, by looking for metabolites that are closely related that have high mass accuracy with relation to that. It then enriches that, okay, so that you get an idea as to which pathways are important. Okay, so we did that for pathway analysis, and this was the sort of one of the mini networks that came out of our enrichment. Okay, so these are things that are positively correlated in red and negatively correlated in green, and the pathways we saw, for example, was tryptophan degradation, okay, increased, okay, vitamin E decreased, okay, and carnitines and acyl carnitines decreased, okay, and you can see very good correlations within those. Now, the problem with Mummichog is basically that it is all like a stack of cards, is that you're basing identifications on accurate mass alone. And of course, we're looking at correlations here, so we must not confuse correlations with causality. So being good analytical chemists, we then went and bought 16 standards. We did LCMS, MS on the Orbitrap, and we matched against those standards. Okay, and the standards that we bought were ones that were representative of things we saw in carnitines, acyl carnitines, tocotrienols, and tryptophan and cunurine and things like this, okay? And of the 16 standards we chose, we identified 15 of them correctly. So we were very happy with that. We thought we better validate this, so we went to wave 6, and we took people who were in the frail and in the resilient characteristic, and then we just measured these 15 standards or 15 metabolites. We just targeted those, okay? And it stacked up exactly how this sort of discovery phase in all of those individuals. We saw that for frailty, that we saw tryptophan degradation, we saw tocotrienols go down and carnitines go down. So, well, we sent that off for publication in Nature Medicine, and we were asked to do some more validation by reviewer 2. Okay, and we'd like to think of reviewer 2 as being related in this way, because we measured another 700 individuals, okay, and we got exactly the same results. We went back to Nature Medicine, and that reviewer still rejected it. And the reason they rejected it is they said you must do this in an independent cohort. Now, given the fact that these 50-year-olds upwards have been followed for 18 years, there really is no other cohort that we could have looked at. So that paper got rejected from there. It went to Nature Comms, and unfortunately the editor decided to chase that reviewer again, and he gave us a little bit of a rough ride, but we eventually got there, and we were very happy with the results. So let's have a look at the links to aging. Okay, so in terms of vitamin E and its analogues, so these tocotrienols, okay, they're there to protect fatty acid groups from lipid peroxidation. Okay, and so with more fatty acids, and remember we see a reduction in fatty acids, and this carnitine shuffle generates and shuffles acetyl-CoA for energy production. So within the frail metabotype, if you like, we have a lower abundance of vitamin E, okay, analogues and carnitines, and this indicates to us that we get a down regulation of this system and basically lower energy output from mitochondria. And it's a little bit like the citric acid story for aging. Okay, so the people who are going into a frail phenotype have this problem with their mitochondria. Okay, with that, I'd like to thank you very much for your attention, and to thank all the people who have been involved in this work. Thanks for listening.
Thanks so much for that. That was wonderful. I was hopeful that, just an initial question. So there's obviously a huge amount, I think, the breath field as a whole can learn from metabolomics. What's your kind of absolute top key bit of advice to the breath community in terms of what we can learn from what's already been learned in metabolomics?
I think a lot of it's already been learned. I mean, the key things in the metabolomics field are identification of metabolites and the fact that you miss loads. So there's some nice papers out there, I think one particular by Peter Dorenstein, which discusses the dark metabolome, which is basically things that, it includes things you can't identify, but also things that you can't currently measure because you need to get them to ionize. So there are going to be lots of things that you don't know about, and this is one of the challenges. So metabolite identification is one. The fact that databases are full of metabolomics data, of which none of those data are quantitative. So learning how to give values where the y-axis means something that's not an ion count would be really valuable.
So one question came through from Yuri: how many times do you split data on training and test sets?
Yeah, so I guess we're lucky in the fact that in both those analyses we collected over 1,000 samples. in fact nearly 2,000 for one of the studies. So we can use bootstrapping, and in bootstrapping you're basically doing you do sampling with replacement. So basically you stick all the samples in the bag, you take one out and you say that's now in the training set, you put it back in the bag and do that again until you've got as many samples in your training set as you had samples. And on average, you select 63.2% of the data and then you test with everything else. In terms of the number of times we do that, we typically do that 1,000 times or even 10,000 times. Computational power is not an issue at that point when it's taken you 6 months to 12 months to actually generate the data in the first place.
So question from Christian here downstone. Are there increased markers of oxidative stress with frailty?
Yeah, that's a good point. We haven't seen those as yet, and a lot of that is because we've not now gone back to the data. So unfortunately, this is one of these cases that project finished. We found those particular markers. We've actually written grants around exactly that question to follow up in other cohorts, but currently the UK research councils don't particularly like it.
I was hoping to ask one more question. So a lot of the work that we've been doing at Alstom recently is where we introduce a non-exogenous probe, what we call the VOC probes, to target a particular pathway. So we're doing a lot there in liver disease and cancer. I was wondering if you had any thoughts about, whether that similar type of approach has been applied in more conventional metabolomics, and if you had any kind of thoughts on, it compared to some of the more untargeted based approaches.
Yeah, so people do both. I mean, you're talking to someone who's started off from the untargeted field. So what we do is when we then discover things, as you saw in the second example, we go and identify them and then quantify them. In terms of other potential solution, and there's Biopsy, for example, will sell you kits that allow you to measure 300 metabolites that cover a wide range of metabolism. And when you see those, you've identified them definitively. So level 1 of Metabolomic Standards Initiative. So there's use for both of those. Obviously, I'd advocate that untargeted means that you can fish a little bit, and you might find pathways that you didn't know would be important. And if you think about the fact that many disease processes are due to imbalance in microflora, so in other words, your microbiome, hence the use of this word superorganism, when you get imbalances in that, that's going to be very difficult to understand unless you target metabolites that are specific to that microbial community.
And as well as some of those targeted analyses, are substrates or probes introduced at high abundance?
So if you're talking about chasing 13C carbon, not so much in people, maybe in animal studies. So maybe I've answered the wrong question there by saying that people do targeted stuff, but people in cell culture will do flux analysis, and it's quite interesting because I'm old enough to remember the early discussions in the metabolomics field, and everyone went, well, flux isn't important, so we'll just do time points. And now the fluxomics community is really quite huge, and realizing that if you want to see pathways, you need to see, substrates and products within those. And that actually, that's one way of looking at it, is chasing 13C or chasing 15N.
I think we've got time for one more question from Alan. By splitting the injection between GC-GCMS and GC-GCFID, quantitation could be observed. Have you attempted this?
That's not a question for me.
Okay.
We just did GCMS. So we did actually have a LECO Pegasus 4, but this was in the early days when the deconvolution was hard. And after having run several hundred samples on it, we found the deconvolution too much of a challenge. So unfortunately, we turned that instrument into a GCMS.
Okay, no problem. Well, thanks a lot, Roy. I really appreciate your time and contribution today. Very appreciative of that.